Maddie, Noha, and Mirodi present DNA's chemical structure.
Have you ever wondered what living organisms were made up of?
Of course you do, but I'm going to tell you anyways.
All living organisms are made up of cells that each contain DNA.
Sounds simple, I know. But there is more.
DNA is a polymer that is made of units called nucleotides. The nucleotides are made up of three different components. One nucleotide has a sugar group, a phosphate group, and a nitrogen base. There are 4 different possible bases: adenine, thymine, guanine, and cytosine. A way that you can use spacing to study DNA is going over the information throughout a long period of time. Let's say you have a test coming up in 3 weeks on DNA. You'd probably tell yourself, oh, I have 3 weeks, I'm fine, and wait until the night before to cram it all in. But if you were to start 3 weeks ahead and go over information each night for about 15 minutes, and then the week of the test start to study for about 40 minutes, and then the night before the test study for 30 minutes, this would prepare you way better than cramming it all in the night before.
The bases on a single strand of DNA act as a code. The letters form 3-letter codons, which code for amino acids. These amino acids are the building blocks of proteins. To understand the importance of DNA, we introduce to you the central dogma. DNA goes through the process of transcription to form a copy of mRNA that goes through the process of translation to ultimately produce proteins. These proteins help our body function. Dual coding involves combining drawing or visual representations in addition to audio or written information. In this video, we demonstrate the repeated use of drawing to help in learning the chemical structure of DNA.
DNA is held together by hydrogen bonds between the nucleotides. The nucleotides are attached to the sugar phosphate group. Adenine always pairs with thymine, and guanine always pairs with cytosine. Retrieval practice is the process of reconstructing concepts and information you already know to solidify that knowledge. The ways to use retrieval practice can be through flashcards, fill-in-the-blank questions, and label diagrams from your memory.
Dual coding and retrieval practice. Using these two together could be very effective. For example, if you were to remember how the DNA structure looked by memory and you could draw it out from memory, then you will see the DNA and start to be able to slowly piece it together how the DNA should be set up fully. Retrieval practice and spacing. This would be studying over a period of time, using flashcards and reviewing your notes and trying to write down everything you remember for a period of time before the test. Spacing and dual coding. Throughout the 3 weeks before an exam, you would draw out important things for the exam and study that over time. This is very effective for STEM majors because biology uses many cycles and patterns that need to be remembered. Putting all 3 of these together is going to be the most effective and give the best results on the test. This is due to the fact that spacing is very effective for long-term memory. Dual coding is very effective for patterns and cycles, and retrieval practice is good for memory of words and meanings. So if you were to be studying DNA, you would know the structure perfectly and the words that all have to do with DNA so well.